me today. I'm going to give you a bit of an overview of using neuroimaging to understand both typical and abnormal brain development. A lot of my work and a big part of what I'll talk about today is a cohort that we've looked at in ADHD, but I'm trying to give you a bit of an overview of two sort of emerging techniques that we've recently used that could be equally applicable both for typical development as well as any other disorder. So next slide, please. So just to give a bit of an overview in terms of, where we've come in terms of understanding the brain, this changes occurred to the brain across the entire lifespan, but from fetal to adolescence is the most dynamic and some would say important part of brain development. And we can even now get the image on the left there is in utero scan of a brain of a fetus. And so since the emergence and many of the advances of MRI, this has really given us unprecedented opportunity to examine and understand how the brain develops. I think it's really important to keep that in mind that, it's only been in the last sort of 40 years that we've had this opportunity. Prior to that, there was some brain imaging techniques, but they used ionizing radiation. And so it was very difficult to use that, particularly in healthy children, as well as when you're trying to do longitudinal data to look at how the brain develops. And so prior to that, what was really known about brain development was done from post-mortem studies, which, for children and adolescents, that would be far between. And you could argue how much it was normal development if they were post-mortem at that age. Next slide, please. All right, so this is just kind of giving you a very brief overview of, on the left, of all of the dynamic processes that are going on during this brain development. There's this synaptogenesis, then there's also this really important competitive elimination of those synapses as the brain's developing and learning new experiences. And as those networks are becoming more wired, they become more myelinated as well. And so across a range of imaging techniques, if you're looking at structure, looking at the white matter and function, as well as other techniques such as EEG, we can get a sense of how the brain is changing across development. However, because there's so many different changes, there is, a number of different trajectories that different parts of the brain take. So on the right, you can see different properties of the brain structure, such as the cortical volume, and that's also made up of the thickness and the surface area, which have different trajectories. When we look at the white matter, the volume increases across that development. But when we look at measures like, say, FA or mean diffusivity, which are metrics of the sort of microstructural properties of that white matter, they also have different trajectories. Next slide, please. And not only do those different properties have different trajectories, but different parts of the brain also have different temporal trajectories of that change. So there's a real complexity to this brain development, which makes it very difficult to get an understanding of what's happening. But it also shows you just, why, I guess, we've got such diversity across different people in terms of their timing of brain development. Next slide, please. All right, so what can imaging tell us then about mental health? Next slide. I think you shouldn't need too much neuroanatomy to be able to understand that there's something severely wrong with those brains in the top row. These are sort of neurological conditions. But when we take a look at psychiatric or behaviorally defined disorders like the bottom row, there's nothing distinct in those brains that we can tell at this stage anyway from a structural MRI scan. And so we need to delve in and look a bit closer at what's going on to understand that brain behavior relationship. Next slide, please. So this is just like a bit of a slide that I like to have that sort of puts in perspective of where imaging fits into understanding disorders, in particular but even just the general brain process. And it sort of fits along sort of halfway between the end phenotype and the genotype at the other end. So here I've got the example of ADHD, but this could be any disorder or any trait. So at the top there, we've got sort of a collection of symptoms for the disorder, but underlying particular symptoms are different basic brain processes. And that might be inhibition or it might be motivation. And underlying those basic brain processes, they're driven by sort of different brain regions or networks of brain regions, such as, say, the prefrontal cortex. But then those different brain regions or networks also have different neural mechanisms and have different neurotransmitters that they operate and different receptors for those neurotransmitters. And they're obviously then determined by your genes. And at any of those stages, from the brain to the neural mechanisms to the genes, can be altered by the environment. And so really this is where I find it helpful to understand where neuroimaging is fitting in, because it's trying to reveal something that's slightly closer to the end phenotype or the end presentation than the genes itself. Next slide. So really we can be using it to understand this brain behavior relationship. I mean, ultimately the goal is to be able to use these imaging techniques to aid and help reduce misdiagnosis, and aid diagnosis in a real clinical setting, but it's not there for many of these disorders at the moment, particularly the psychiatric ones. It might help identify precursors for onset or help predict treatment response. And some of these are happening at a research level, on a large group level, but not yet on an individual basis. But what we can do is understand a little bit more about the neurobiology. Are these disorders a sort of distinct identity, or do they lie somewhere on a dimension of normality? Is it particular structures or different networks that are involved? Is it within the grey matter, the white matter, or different neurochemicals? What contributes towards the functional deficits that we see in disorders? And is there an effect, either functionally or long-term structurally, of medication? Next slide. I will, as I mentioned, talk a little bit about ADHD in this cohort. So just to give a brief introduction, so ADHD is a neurodevelopmental disorder where participants exhibit inappropriate levels of either inattention and or hyperactivity and impulsivity. This affects around 5% of school-aged children globally. That decreases a little bit into adulthood. But there is this bit of a difference in the sex ratio. If you're looking in clinically defined groups, it's up to about 5 times higher in males. But if you look representatively in a community, it is much smaller. It's 2 to 3 times higher in males. And what this really highlights is that a lot of females are either getting, not referred to clinic or missed in those cases. But ADHD actually affects a wide range of outcomes for these children, academically, cognitively, socially, as well as broader mental health functioning. And many of the impairments do persist into adulthood. Even if the disorder remits officially, many of the functional impairments still persist. Next slide, please. The other tricky thing when trying to look at ADHD, but this is quite common across psychiatric disorders, is that very heterogeneous in their presentation. So they can have different sort of clinical subtypes or presentations, different comorbidities that are quite common, in fact more common than the exception. They can also have different cognitive profiles in the areas that they have deficits in, and they can respond differently to treatments as well. And so on the left, we've got there the 2 different groups in 2 different colours, and a lot of researchers originally defined their categorical group differences. Look, here's an ADHD group, here's a typical control group. But more recently in cross psychiatry, there's been a shift to look, when you see on the right-hand side, across the spectrum of a particular trait across the population that you're looking at. Next slide, please. So this is the cohort that I'll be presenting on today, the data that we've been collecting. It's called the Neuroimaging of the Children's Attention Project. This was a longitudinal community-based cohort where we conducted multimodal MRI. And along with that, we had really extremely well phenotypic data. So we had information from the child, from their parent, from their teacher, as well as direct assessment with the child. And we assessed a range of key functional outcomes, mental health, academic, social, and cognitive functioning, across 3 time points with 18-month intervals. So we collected them from around the age of 9 through to 14. On the right-hand side, you can see the sort of spread of time points and ages that we have across the data. And I say multimodal imaging, we collected structural T1, T2, QSM, which is an ion mapping type technique, as well as diffusion to look at the white matter, as well as resting state functional connectivity. Next slide, please. Just to briefly touch on that, if you haven't had experience with MRI before, it is extremely susceptible to motion, and it is a very challenging environment to work with children, but also children that move a lot. So we put a great deal of effort into getting the children engaged in the study, really wanting to participate. You know, they got a certificate when they came, they got to watch movies for most of the scans, and really this was done for 2 main reasons. One, to reduce the anxiety of for the children going in the scanner. You know, this is a very sort of medical environment and it can be quite intimidating for children. We wanted to have success with them, and not just once, but to come back multiple times. But the other reason was to minimize that motion. So we also have a mock scanner, which is a full shell of an old scanner without the magnet, which we can run a training session within first so that it gets them familiar with the environment. Next slide, please. So that's just an overview of the sort of cohort. I'm going to give a little bit of in-depth talk now about 2 main studies that we've recently worked on or currently have under review. One looking at actually incorporating multiple imaging modalities together, and the other one looking at specific white matter properties. Next slide, please. So when we look at the imaging literature within ADHD, there's actually been quite a lot of imaging studies, and a lot of the very early ones, you can see there on the top right, really focused on volumetric type analysis and looking at the basal ganglia structures and the prefrontal cortex. And this was because of, you know, hypothesis driven that, you know, the behaviors and the deficits that we see in these children are likely to have arisen from dopaminergic systems in frontostriatal regions. And so that's really where the research was targeted at. And then a number of functional studies would also probe these tasks. And we started to realize, though, that it's actually more than prefrontal and subcortical structures. There's also the parietal cortex is heavily involved, as well as cerebellum, and actually a number of regions. And so across all these studies, there is some consistencies, but there's also an extreme lot of heterogeneity. And we started to realize that, you know, we haven't been able to stumble across this 1 or 2, you know, key areas of the brain that are responsible for ADHD, but it seems to be much more a distributed network of regions that is dysfunctional. And so approaches have tried to evolve to that, to look at networks in the brain, you know, across the brain, but also specific functional and structural networks. Next slide, please. So I mentioned right at the very start, when the brain's developing, that, you know, we understand that now that different parts of the brain have both different spatial and temporal time trajectories, you know, in how they're developing. But advances now in statistical analysis allow us to model how these patterns co-vary across different imaging modalities. Next slide. So this technique taps into something called ICA, or independent component analysis, and this is not something new. It's not something new outside of imaging as well. This has been, you know, something from signal processing analyses that have been around for a very long time. And this is something that the brain, in fact, is very good at. This scenario just gives an example of when you're in that sort of cocktail party type atmosphere, and you actually have a lot of signals that are coming into your brain to process, even if just thinking about the auditory sense, you've got your piano player there and your singer, you've got different groups of people having different conversations. But your brain is able to very quickly and with some ease dissociate those different signals and focus in on a particular conversation, realize where, based off spatial and temporal information, as well as other things like the frequency, pass out different parts of that signal. So next slide, please. So this has been used in imaging even for a little while. This is not new, and it started off being used to be able to try and pick up artifacts within the data and where you can extract that and throw it away from the data. But then more recently, it's been used for resting state functional connectivity. So if you're not familiar with this, with with functional MRI, you can look at blood flow within the brain and see what parts of the brain are being activated. In resting state, they're actually not doing a task, but you can see based off the spatial and temporal patterns, which parts of the brain have the same signal time course. And so even though they're not doing a task, those networks you can see at the bottom of the page there, you can still pull out motor and visual and cognitive networks, even though the individuals aren't doing a particular task in the scanner. Next slide, please. So taking that property forward, now we can, new advances have been able to allow us to do this across imaging techniques. And this process is called linked ICA. So this example here has taken diffusion scans as well as structural data on the grey, the white matter, as well as cortical thickness measures. Next slide, please. So this is the approach that we took on our data. We had volumetric data as well as data on the cortical thickness and the surface area. And we also had diffusion metrics, which look at the white matter. So we had our FA maps, fractional isotropy, as well as MD, mean diffusivity maps. So we took all this data from all of our subjects, and we put this into a linked ICA analysis. And from this, we were able to extract 25 spatially independent components that covaried together. And each component weights describe the extent to which that pattern is linked or expressed within an individual. And the bar graph that you can see in the bottom right there shows you sort of each of those 25 components and the sort of distribution of how much of that component was driven, for example, by volume, or others by, say, surface area. But there are some that are fairly heavily exclusively driven by one modality or the other, but then most of them are fairly well distributed. So this was the first step. This is just trying to identify spatial patterns of the brain across different imaging modalities that covaried, and that was the first step. So the next slide, please. Next slide, please. Yep, thanks. So then we took a heap of our phenotypic data, and this is sort of broadly categorized into individual factors like their age, weight, pubertal status, clinical factors, so obviously ADHD symptoms, but also other comorbidities of externalizing and internalizing disorders, autism symptoms, irritability, for example. We also had a range of cognitive factors such as IQ, their academic functioning, spatial working memory tasks, inhibition, sustained working memory. We also had some perinatal factors, so these are retrospective data that we had, but their birth weight, whether they spent time in a neonatal intensive care unit, or whether the mum smoked or drank alcohol during pregnancy, as well as a range of family factors such as the parental education, parenting styles, family quality of life, things like that. So we had all of these brain networks that had covariation, We had 44 different phenotypic variables, and we put them all together to look for multivariate association. Next slide, please. So this canonical correlation analysis looked for multivariate association between the subject's phenotypic data and their imaging features. And what this analysis revealed was 4 independent multivariate relationships. And we loosely categorized them into being driven by head size, development, ADHD, and cognition. And I'll go into those in detail over the next couple of slides. So next slide, please. So the first component that explained the most variance in it was intracranial volume, and that wasn't actually terribly surprising and not particularly interesting either. You can see there, so I'll just explain these images because these is what you see for each component. So we've got 5 rows there. The top row is the brain volume, followed by the 2 rows of diffusion values for looking at white matter, and then followed by cortical thickness and surface area. So this marker of head size was heavily driven by that intracranial volume. That's why that top row is almost all yellow. That's an increase in head size on the volume measure, as well as the bottom one you can see is surface area as well of that cortex. So as I said, not particularly surprising, but quite nicely, we did also see that that marker was associated with other things that we know to be associated with head size, such as being male, generally have larger head sizes, better cognitive performance, better socioeconomic status, and a higher birth weight as well. Next slide, please. This next marker we defined as development. So this is anything that's independent of head size again. And we can see here now we've got different patterns of both increase and decreases of volume, white matter diffusion, as well as cortical thickness and surface area. And so this particular pattern of increases and decreases was associated mostly with their weight, pubertal status, and age, as well as you're more likely, less likely to be on medication. Next slide, please. All right, so this one was our more interesting one because this was, head size and development aside. This was specific to ADHD phenotype. So this particular pattern of increases and decreases across those imaging modalities, you're more likely to be male, older, but of younger pubertal stage, higher impulsive symptoms. Inattention was still significant, but it didn't reach 0.001, which was the cutoff for our graphs here. You're more likely to have autistic symptoms. You're more likely to be medicated, more likely to have had stressful life events, less consistent parenting and less family quality of life, as well as you're more likely to have spent time in a neonatal intensive care unit as a baby. Next slide, please. And then finally, this was our marker of cognition. So this is aside from the cognitive deficits that were picked up in the ADHD component, this was independent of that. So this was a profile that you're more likely to be male, higher irritability, which is interesting because irritability is a trait which is often seen as comorbid within ADHD. But here we see this irritability, independent of ADHD. So you're greater irritability but less hyperactive, as well as poorer academic language, visuospatial reasoning, cognitive flexibility, and also your mother was more likely to smoke during pregnancy. Next slide, please. I won't go into the details of this, but basically we did a range of robustness testing, the different number of components we took, and that type of thing, just to show the robustness of the findings. Next slide, please. All right, so, that was really cool that we found these different patterns of development, head size, ADHD, and cognition. And even though that we came into this wanting to look at this dimensionally and, not categorically, we wanted to see, well, is it enough to distinguish categorically? And so we actually found that our marker of ADHD was significant and was significantly able to dissociate ADHD from controls. The effect size is not huge, admittedly, but it was able to pick that up and it did not find differences for head size, development, and cognition. Next slide, please. So that was pretty cool. We found that in our own data, but we wanted to see, well, can we take what we're finding here about these brain development patterns and predict ADHD in a different group? So we took data from the ADHD 200 cohort, which is a publicly available data set. Now, this data set didn't have all of the multimodal imaging and didn't have all of the phenotypic data, but it did have structural data and it did have, small bits of phenotypic data on their ADHD symptoms. Next slide, please. So taking just our volumetric components, we were still able to predict in the external cohort. So our marker of head size was able to predict intracranial volume in the independent cohort. Our marker of development was able to predict age within the independent cohort, but importantly, that component didn't predict ADHD. Our ADHD marker was able to predict both hyperactivity as well as inattentive symptoms, but importantly not age or IQ. And our cognitive marker didn't predict anything in that cohort, but they didn't have cognitive measures other than IQ, and IQ wasn't one of the significant ones from that. So yeah, each of our markers was able to predict the corresponding phenotype across an independent cohort, which was nice. Next slide, please. So yeah, if you're interested in finding out a little bit more about that particular project, that was published last year, not 2019 now, in American Journal of Psychiatry. I've got the reference there. You can have a look. But yeah, basically we used data-driven multimodal imaging here with lots of phenotypic data, and we're able to dissociate these novel sets of imaging patterns, which were predictive of phenotypes within an independent cohort. Next slide, please. So I'm going to change gear now and talk a little bit more about white matter tracking. We had taken that large multimodal approach and we used diffusion tensor imaging in that, which I'll get to in this next bit. But we really wanted to delve into the white matter a little bit more. White matter is the white matter connections that convey information as part of these structural networks. And so we want to find out a little bit more about the biological properties of this white matter. Next slide, please. So I'll just give a brief overview first off of white matter imaging in case you're not familiar with it, because we've started to use a relatively new technique here. So diffusion weighted imaging is an MRI technique that's sensitive to water in the brain and how water moves. And so it can differentiate between different tissues based on the diffusion of water within that tissue. And this allows us to infer some of the architectural properties of that white matter. You can see that picture at the bottom there on the left-hand side is some tractography that's generated from diffusion data versus the histology type dissections of a brain, a real brain. Next slide, please. So how is this measured? Basically, using Brownian motion, the idea that if a molecule of water has no obstruction, it will diffuse randomly and equally in all directions. And so if you look at that example on the left, you've got that red circle, which is the molecule of water. It can move around equally in all directions. And so if you took a probability of the directions of diffusion, or if you averaged across a whole bunch of water molecules, you would get a very soccer ball shaped sphere of possible diffusion directions. However, if that molecule of water, that's it, thanks. If that molecule of water is restricted somehow, such as being in a straw or something like that, then it will have an uneven distribution of potential diffusion directions, and you end up with something much more like an Aussie rules football shape. All right, we'll go on to the next slide, please. So the extent to which water can move around or is impeded is dependent on a number of sort of underlying neurobiological factors. And these aren't limited to, but can include things like the fiber bundle density, the axon diameter, whether that axon is myelinated or how thick that myelin is, how permeable the membranes are, sort of psychoarchitectural properties within the axon, as well as macrostructural ones about whether those axons are branching or crossing. And so most of the diffusion imaging that has been done to date has used something called the diffusion tensor model, and DTI is the acronym. And if you've read anything to do with white matter imaging, you will be familiar with this term. But DTI is not the same as the actual acquisition of the data. It's a model used to model what's going on with the diffusion within that data. Now, diffusion tensor imaging has a number of two different problems, really, and I'll sort of talk about them over the next couple of slides. But in terms of trying to understand these underlying microstructural properties, DTI is really sensitive to some changes within these properties, but it's not specific to any particular one. So looking for a change in one of the DTI metrics like FA, fractional isotropy, or mean diffusivity, doesn't actually reveal what specific neurobiological property is changing underneath. Next slide, please. Just to highlight some of these interpretation troubles that we can get into, we look across the top row. First on the left-hand side, you've got a bundle of fibers, and you've got your tensor modeling of that fiber direction. If there are less fibers, for example, in the next, right in the middle of the page, you've got this sort of less directional, or you'll have a lower FA, or you might have the same number of fibers right on the right there, but they might be unmyelinated or lower diameter, and you would also see a similar decrease in FA. And these properties kind of make sense. Oops, sorry, just go back first. These properties kind of make sense, but only really when the fibers within a voxel are all pointing in the same direction. And other than sort of the middle of the corpus callosum, most of the fibers across the brain, in fact 90% of the brain, voxels have crossing fibers. And so if we look at the example on the bottom there, we can get this sort of paradoxical type of information, particularly when we're looking in a clinical group. So an example of the bottom left there, where you've got these crossing fiber bundles, you also have a lower FA compared to the one at the top, even though it doesn't speak to the integrity, if you like, of those two crossing fiber bundles. But the mere fact that the diffusion direction is kind of averaged across two different directions. However, sorry, just we'll go back one sec, just for that last bit of the slide in the middle down the bottom there. If you then do have, in say, a disorder, one of those crossing fiber bundles is something wrong with it, or there's demyelination or cell loss or something like that, you can end up with a higher FA, paradoxically, in a clinical disorder. And so it can be more difficult to interpret what's going on underneath, but it also has issues for conducting the tractography to begin with, to identify the correct tracks. Next slide, please. All right, so there's been a number of attempts to try and improve upon the diffusion tensor model. And this one I'm talking about in particular is called CSD, or constrained spherical deconvolution. And looking on the left there, you can see these examples in green of the fiber, hypothetical fiber directions within a voxel. Underneath that, you can see that the orientation of those fibers works quite well for the, for the vertical ones and the horizontal ones. As soon as you have those two fiber populations crossing within a voxel, you end up with a averaged tensor within there, and it gives you little directional information within that. And so it's a shame I can't use the pointer to really show you, but On the sort of right-hand side there, on the left of that, where you can see the yellow shapes, this is modelling a section of the brain where you do have really bad, if you like, crossing fibres. You've got the corpus callosum fibres coming through left to right. You've got the cortical spinal tract coming up, and then through the page you also have the superior longitudinal fasciculus. And so in conducting tractography, you take these football shapes and you go voxel to voxel to try and track where that fibre is going. However, what happens when you get those spheres in the middle of those crossing fibres, it causes errors within the tractography. It might track along and either stop as soon as it hits something that without a direction, or it can be misleading and actually take the wrong path up that tractography. And you can see that in the example on the bottom there, where you've got in yellow there, you've got the corpus callosum fibres, and in red you've got the corticospinal tract. So in the top example down the bottom there, you have this real limitation with the projections coming out from the corpus callosum. As soon as that tractography hits that area of crossing fibres, it's losing that track and is only following them up the top. And so you're missing out on that big spanning of fibres that you can see down the bottom using the tensor model. So CSD ends up with having different lobes for different directions. And so even when you're in areas of crossing fibres, you end up retaining directional information. And so it's a bit hard to see, but on the far right there, you've got that same brain region with that lobe information, and it's much easier to follow tractography through and identify tracks of consistently. Next slide, please. So here you can see that, sorry, I'm kind of jumped ahead here. All right, we'll skip just to the next slide, please. So basically, those constrained spherical deconvolution has been around for a little while, a number of years now, and that improves our tractography. But recently, we can use that directional information within a voxel to get fibre-specific metrics, and it's so-called fixel. And so that's fibres within a voxel. And this also helps to reveal some more biologically plausible properties. As metrics, we can look to understand that underlying white matter microstructure. So we can get measures of fibre density as well as fibre cross-section. So fibre density is that packing of axons, and that might change due to, say, axon loss or to myelination, or fibre cross-section is a much more macrostructural property. So there might be atrophy, or when we're looking at development, might be growth. But the image down the bottom, you can actually see in that blow-up square, it's following in yellow is your significant fixels. So within a voxel, you can actually see the yellow is being significant, showing this corticospinal tract difference, but the crossing fibre within that fixel, in that voxel, sorry, is not showing a difference within the fibre density. Next slide, please. So if you're interested in finding out more about this particular technique called fixel-based analysis, I've just been part of a team that's just done a comprehensive review on this technique. It's only been around for a couple of years now, and that you can see on the right-hand side there, the number of publications using this technique is really sort of increasing. There was 57 papers now we included as part of this review, and they were in different areas of brain injury, development, adult, psychiatric, normal development. But also not only reviews the data within those studies, but also presents a lot of the interpretation and some of the challenges from that interpretation too. So it's currently under review, but we do have the preprint. I've got the address down the bottom there. You're welcome to access in the Open Science Framework. Next slide, please. So basically we've taken this approach for the first time to try and understand the white matter properties within ADHD. Next slide. I'll try and skip over a few of these quickly because I know I'm running a bit slow on time. So this study, we used 144 children aged 9 to 11. This is not the longitudinal data. This is just the baseline data, the first time point. So here, as you can see our scanner at the Children's Hospital. We used a Hardy sequence, which is a high B value, high direction sequence. Next slide, please. I won't go into the details of this one, but this sort of just highlights the processing steps here. But one of the important things to highlight is that we actually used a template, a cohort-specific template based off the tensor values within the white matter metrics to minimise biases in registration. Next slide, please. We took an approach to selecting our tracks, and this is a fairly recent program as well called TRACT, which is available, and it actually uses machine learning on those lobes to generate consistent, reliable tracks across the brain. It's really quite nice. So these are the tracks that we used. It's a collection of association fibres, projection fibres, and what am I missing? Callosal fibres as well. Next slide, please. So taking all these fibre bundles, we looked at what was differences between ADHD and control within the fibre density and fibre cross-section. So we actually found lower fibre density, particularly within the corticospinal tract, the frontopontine tract, as well as the inferior frontal occipital fasciculus. So in these regions, ADHD presented with lower fibre density. Next slide. Similarly with fibre cross-section, so quite consistent was that corticospinal tract had lower, so this is with the fibre bundle size, as well as the parietal occipital pontine tract, as well as the uncinate fasciculus were all lower within ADHD group. Next slide, please. So we also took this sort of, using these same tracks, looked at FA, which is the tensor metric, to look at how consistent that was with the literature and with these newer metrics. And we actually get a bit of a mix of results here. So, the findings of the inferior longitudinal fasciculus, the inferior frontal occipital fasciculus, and the corpus callosum, they are actually quite consistent with findings from FA. Within the tensor imaging, but then they're also, in terms of the two bottom brains, fairly consistent with the fibre density and cross-section measures we got. Next slide, please. So that was looking at the groups as categorically different groups, but we also looked dimensionally with symptom severity, and then we found a fairly consistent pattern, although this one was the only one that was significant, showing that as ADHD symptoms increased, fibre density decreased. And in particular in this frontopontine tract, and if you look at that small brain down the bottom, you can actually see, even though these fibres come up the midbrain and go to the entire prefrontal cortex, this really was restricted to part of the fibres going up next to the motor cortex, the supplementary motor cortex area, which is an area that has been implicated quite a lot within ADHD and also within inhibitory control in general. Next slide, please. So this was the first comprehensive characterization of white matter using this pixel-based analysis in ADHD. We demonstrated lower fibre density as well as fibre cross-section in a range of association and projection pathways that are known to be critical for a lot of the higher order behavioural control and motor functions that we see in ADHD. This CSD model clarifies particular inconsistencies we find within the findings. The paper goes into some of the inconsistencies that we've seen across DTI and how now being able to resolve some of these crossing fibres helps to resolve that. And really adds it for a case of moving beyond the tensor model. And our future work will this will then start to track that with the longitudinal data that we've got. Next slide, please. I'm just going to very briefly now just touch on a couple of the other projects, other applications that we've got going with the data. So in the top left-hand corner, using that same analysis technique with pixel-based analysis, but looking at pubertal development longitudinally, just typical development, and how fibre density changes within tracts across development. The next bit along, we've got how longitudinally how those aspects of development change and how mental health changes with that. Then in the top right-hand corner there, this is not actually including brain imaging data yet, but this is looking at their cognitive trajectories over time. So this is looking at their sustained attention performance, and to how that is consistently worse in ADHD, even if they remit out of their ADHD. Phoebe, who's a PhD student who's working on that at the moment, is currently looking at both the resting state and diffusion data trajectories associated with sustained attention. Then down the bottom there, we've got a number of studies looking at pubertal development in the brain, the effects that puberty has on that brain development. This is looking both at the cortical thickness as well as subcortical volumes, both how their pubertal stage as well as hormones influence those trajectories of brain development. Next slide. So what next? So part of this data as well, we're contributing to the Enigma Consortium, which is an international consortium. We're combining imaging as well as some genetic data across different cohorts across the world, and producing some really nice findings in different areas. We also have some genetic and epigenetic data on these cohorts, which we'll be integrating into our analyses. Next slide, please. I wasn't going to go into too much detail about this, but just to highlight it as an emerging technique that's been used, quantitative susceptibility mapping. This is a way of mapping iron content. Now, iron content mapping in itself is not necessarily new, but this is the most recent and most specific to iron that we've had so far. And this has been used a lot, iron mapping techniques have been used a lot in degenerative disorders like Parkinson's and Alzheimer's, because of the high oxidative stress that you have with iron content. But we're using it in this context because iron and dopamine have a really important relationship, and iron is found to increase across development in the developing brain, particularly in those subcortical regions that are high in dopamine, and dopamine being the main hypothesis-driven approach for ADHD, as well as the main neurotransmitter that medication works on. It means we're probing these areas of the brain to look at how they develop over adolescence as well. Next slide, please. I just want to acknowledge there's a huge team that has helped to collect this data as well as process it. In particular, I want to highlight Ian Filcher, who is the postdoc who conducted the pixel-based analysis, and Gareth Ball, who conducted the multimodal imaging analysis. And we've got a picture of our lab there in December this year. It was the first time we'd been face-to-face because we'd all been in lockdown since March, so it was nice to have a get-together. And that's it. I think there's one more slide just to say thank you, and I'll open the floor for any questions you might have. Thank you.